Hello, we are Group 17. We are going to talk about minor histocompatibility antigens. In this issue, we will first explain what minor histocompatibility antigens are and then how they are related to reactions using organ transplantation as an example. First, let's talk about what minor histocompatibility antigens are. Minor histocompatibility antigens, also known as MIHA, are peptides that are present on the surface of cells of donated organs, mainly in organ transplants. This also relates to the immune response to organ transplantation. Here, a similarity to this is major histocompatibility antigen, MHC antigen. It forms the major histocompatibility gene complex MHC by binding to the major histocompatibility gene complex molecules. MHC in humans are known as human leukemic cell antigens HLA. The rejection that occurs in organ transplantation occurs when this HLA is different between the donor and recipient and T cells recognize it as non-self. However, rejection can occur despite this HLA match. This is where minor histocompatibility antigens come into play. If the amino acid sequence of the MIHA that binds to this HLA is different, it is recognized by the T cells as non-self, resulting in minor rejection. Next, how MIHA is involved in the response is explained using the example of organ transplant rejection. Firstly, MIHA is a diverse peptide, and this peptide may differ from individual to individual. So why do these differences arise? Reasons include single nucleotide changes, SNPs, and coding regions of genes, gene relations, frame shift mutations and insertions. This article describes the reaction mechanism when an SNP occurs. This SNP results in different amino acid sequences between the donor and the recipient when translated into different amino acids. This amino acid sequence is broken down into short peptides by the proteasome. This peptide, which later becomes MIHA, is then transported to the endoplasmic reticulum by a peptide transport pump called TAP. There, the peptide binds to HLA molecules and is presented to the cell surface through the Golgi apparatus. However, this MIHA is different between donor and recipient and is therefore recognized as non-self by the donor's T cells, which initiates an immune response, resulting in rejection. Major minor histocompatibility antigens include the HY antigen, encoded by the Y chromosome, and histocompatibility antigen 1, HA1. Some HY antigens in particular are universally expressed in nucleated male cells and are associated with rejection in male recipients and female donors with matching HLA.